Welcome back. Today we discussed about the chapter Sexual Reproduction in Flowering Plants. Plus 2 Botany, second chapter. Angiosperm is known as flowering plants. In this picture, you can see sepal, petal, stamen, anther, pistil, ovary, ovule, etc. These are a structure present in flower. Here the sepal and petal is known as non-essential whorls because they do not directly participate in sexual reproduction, while stamen pistil ovary portion is known as essential whorls because they directly participated in sexual reproduction. There are three events taken place in the reproduction of flower. They are pre-fertilization, fertilization, and post-fertilizational changes. In pre-fertilizational structures and events, you can see or you can study stamen, microsporangium, pollen grain, microsporogenesis, pistil, megasporangium, embryo sac, then ovule, megasporogenesis, etc. Then pollination, kinds of pollination and agents of pollination. Then economic importance of pollen grains, pollen pistil interaction, and finally artificial hybridization. In fertilization topic, few topics are to be discussed. They are double fertilization and triple fusion, then primary endosperm nucleus, then zygote, then post fertilizational changes. You can study endosperm, embryo, seed, fruit, apomixis, and polyembryony. Event 1, pre-fertilizational structures and event. In this picture you can see a flower that carry male and female reproductive structures and a corolla. This is a typical structure of stamen. In the first figure you can see a filament and an anther. These filament and anther together known as stamen. Then in the next figure, second figure, you can see the pollen sac. There are 4 pollen sac you can observe here. A typical angiosperm anther is bilobed and each lobe has a theca, dithesis. So anther is 4-sided tetragonal structure consisting of 4 microsporangia. Microsporangia later develops into pollen sac. In this picture, the second picture, you can see the pollen sac, that is 4 pollen sac can be observed. There is a dot at the center and that portion is known as connective, through which the filament transport the nutrients for the anther. Here it is a structure of anther. Here you can see epidermis, endothelium, middle layer and tapetum, and they can be discussed later. Here in this picture you can also see a structure called stomium. This stomium has hygroscopic ability to absorb water from the surroundings so that it gets swell when the anther matures and these pollen grains get escaped through the dehiscent portion of stomium. This is an enlarged portion of one side of anther. In this picture there are 3 layers you can observe. The first layer is epidermis and second layer is endothelium and the third layer is middle layer and fourth one is tapetum. The first three layers, that is epidermis, endothelium, middle layer, serves as protection in function and tapetum functions as nourishment. That means it nourishes the developing pollen grains. That is microspore mother cell get nourishes and they get divide meiotically. The next topic is microsporogenesis. Sporogenous tissue, that is pollen mother cell. In this picture you can see the microspore mother cell. This is known as sporogenous tissue. These sporogenous tissues undergoes meiotic division and produce microspore tetrad. Microspore tetrad means four spores together. That you already understood that due to meiosis, four spores are produced and they are haploid in nature. And this microspore tetrad later separates and matures to pollen grain. That is the process of formation of microspore from a pollen mother cell, microspore mother cell through meiosis is called microsporogenesis. Remember, the process of formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. This haploid microspore later metamorphose into pollen grain. This is a structure of pollen grain. It has two layers, that is exine and intine. In the first figure you can see the exine and intine. Exine has some ornamentation and also exine is made up of sporopollenin. Remember, exine is made up of sporopollenin and intine is thin and made up of cellulose. Here in the second figure, exine, intine, there are two nucleus can be observed, that is vegetative nucleus and generative nucleus. Vegetative nucleus and generative nucleus case can be studied later. Here in the first figure you can also see a germ pore. Germ pore means there is no sporopollenin present at that places. Through this germ pore, the nucleus inside the pollen grain can emerges out. This sporopollenin is the most resistant material as it can withstand cold, hot and no enzyme can't be digest it. That means this sporopollenin, a material that made the exine of pollen grains, is the most resistant material so that the pollen grain can withstand in cold, hot and extreme conditions. In some areas of exine they lack sporopollenin through which the pollen tube can emerges out and they are called germ pores and intine are thinner. You can again see the same image, that is pollen grain as a whole and section of pollen grain. A pollen grain consists of two cells, that is vegetative nucleus or tube nucleus and generative nucleus. Vegetative cells are large, then abundant food reserves are present in it. while generative nucleus is small and float in vegetative cell and it is spindle shaped. About 60% of pollen grain contain two cells and remaining pollen grains in other some plant species contain three cells. That is, vegetative nucleus act as a protruding structure. That means it acts as to produce a pollen tube while germinating, and generative cell during its transport it splits and produce two male gametes. It can be discussed in the later portions. Then the pistil, megasporangium and embryo sac. Gynecium represent the female reproductive structure. The pistil may be monocarpous, that means only one carpel, or bicarpellary, means two carpels, or multicarpellary, that means many carpels. Then if the carpels are free, then it is apocarpus, and it is fused, it is syncarpus. This topic that we studied in the plus one topic, that is morphology of flowering plants. Pistil consists of style, stigma and ovary. Here the stigma is used as the landing site for pollen grain, and style is a slender tube which leads to ovary. The ovary contain ovules that can be discussed in the later portions. Here it is a structure of ovule. In this structure you can see the funicle, that means stalk of ovule, then hilum, raphe, etc. The body of ovule consists of two ends mainly, that is chalazel end and micropylar end. Micropylar end is open through which pollen tube entered into the embryo sac. And you can see in this picture nucellus. Nucellus means many cells. Then antipodal cells. Antipodal cells. In this picture you can see three cells are antipodal, situated in the chalazel end. Then polar nuclei, not pollen nucleus, polar nuclei at the center, or it is otherwise called central nucleus. Then at the bottom a large cell called egg at the center, and the two supporting cell is known as synergids. Then there are two protective layers, that is outer integument and inner integument. So we can say an ovule is the embryo sac covered with outer and inner integuments. Ovule. Each ovule has 1 to 2 protective envelopes called integuments. The nucellus fill inside the integument. There are different types of ovules present. Here are some ovules. The first one is orthotropous, pointing upward. Second one is anatropous. Third one is camphylotropous, and fourth one is amphitropous, and fifth one is hemianatropous. Among these ovules, the B type, that is anatropous ovule, is the typical ovule. Typical ovule means most of the plants contain anatropous ovule. Then megasporogenesis. Like microsporogenesis, plants also show megasporogenesis, that is the formation of megaspore from megaspore mother cell, that is commonly known as MMC, through meiosis is known as megasporogenesis. Once again, formation of megaspore from megaspore mother cells through meiosis is known as megasporogenesis. From a megaspore mother cell, four megaspores are produced through meiosis. Among them, three of them get degenerated and one become functional. And the functional megaspore get repeatedly divided in mitosis three times and produce eight celled condition, and each cell arranged in embryo sac in a special manner. Such a process is known as monosporic embryo sac development. This process can be discussed using a diagram. Here, the megasporocyte, that means megaspore mother cell, its ploidity is 2n and it divides first as meiosis 1 and meiosis 2. That means after meiosis, from a megaspore mother cells, four haploid megaspores are produced. Among them, three megaspores undergo degeneration and one become functional megaspore. These functional megaspores undergoes mitosis, first mitosis, second mitosis and third mitosis, and it results the formation of eight cells, and these eight cells are arranged in a special manner in the embryo sac, that is three at the top, that means in chalazal end, three at the bottom, that means micropylar end, and two join together and stay at the center, and each of them have special names and they can be discussed in the next slide. The three at the chalazal end is known as antipodal cells, and the central cells is known as polar nuclei. Polar nuclei's ploidity is 2n condition, and the micropylar end three nucleus are present, and the center cell is known as egg. It is larger than others, and egg is supported by synergid cells. So a typical embryo sac has 8 nucleus while it has only 7 cells, because there are 3 antipodals plus 2 polar nuclei plus 2 synergid plus egg, so 8 nuclei present, while in the case of polar nuclei or central cell, they join together and act as a single cell. So total cell counting 3 at the 3 at the chalazal end, 3 at the micropylar end, and 1 at the center, so 7 celled condition. So a typical embryo sac is 7 celled while 8 nucleated. Then pollination. Pollination means transfer of pollen grains from anther to stigma is termed as pollination. In this picture you can see pollen grains collected by the honeybee is transferred to the stigma. Pollen grains is comes from the anther and collected by honeybee, and it is deposited over the stigma of the pistil. There are two types of pollination, that means self pollination and cross pollination. In self pollination, pollen grains from the same plant reaches to stigma. In cross pollination, pollen grains from one plant transferred to another plant's stigma. That means they are genetically different in nature in the case of cross pollination, while in the case of self pollination the same plant pollination occurs. In self pollination there are two categories, that means autogamy and geitonogamy, and in the case of cross pollination xenogamy is occurred. Autogamy, transfer of pollen from anther to stigma of same flower in same plant. That means there are two types of flowers present. One is chasmogamous flower and another is cleistogamous flower. In chasmogamous flower, which open all its parts, that is, they are exposed flowers. Example, hibiscus, rose, etc. While in the case of cleistogamous flower, these flower don't expose its parts. Example, pea plant. And cleistogamous flower generally show autogamy. That can be discussed later. That means autogamy means transfer of pollen grains from anther to stigma of same flower in same plant. Here, this picture represents a chasmogamous flower. Here you can see anther, stigma which are exposed, while in this picture you can't see anther and stigma etc. because they are covered by the corolla itself. So they are cleistogamous, don't exposing flower. In this kind of flower, this autogamy is taken place because only self-pollination can taken place in this kind of flower. Then the next case is geitonogamy. In geitonogamy, transfer of pollen grain from anther to stigma of different flower in same plant occurs. That means in autogamy and geitonogamy, both the plants are same. That means autogamy and geitonogamy belongs to self-pollination. While in the case of xenogamy, here transfer of pollen grains from anther to stigma of different flowers in different plants taken place. That means two different plants participated in this xenogamy. So it is an example for cross pollination. Here we can easily understood the topics, that is first one is autogamy. Here pollen transfer within one flower in one plant, so it is self-pollination. And in geitonogamy also pollen transfer between flowers of one plant individual, while the two different flowers are participated. It is also genetically both the two flowers are genetically identical because they are in the same plant. So it is also an example for self-pollination. While in the case of xenogamy, you can see pollen transfer between flowers of different plant individual. That means two flowers in two different plants, so they are genetically different. So such a kind of pollination is known as xenogamy, and xenogamy belongs to cross pollination. Then the next topic is agents of pollination. There are two agents mainly, that is abiotic and biotic. Abiotic means non-living agents like air and water. Biotic means living agents like and insect, animals, etc. And pollinating agents are broadly classified as abiotic agents and biotic agents. Example for abiotic agents is anemophily, hydrophily. And examples for biotic agents are entomophily, ornithophily, chiropterophily, malacophily, myrmecophily, etc. All these are belongs to zoophily. Zoophily means transfer of pollen grains by the help of animal. Here, water transfer of pollen grain is known as hydrophily. Air transfer is known as anemophily. Insect transfer is known as entomophily. Bat transfer is known as chiropterophily. Animal transfer is known as zoophily. Bird transfer is known as ornithophily. Snail transfer is known as malacophily. Ant transfer is known as myrmecophily. And wind pollination is more and water pollination is about 30% and in among monocotyledon. That means wind pollination taken place more in the case of monocotyledon. Example, Vallisneria, Hydrilla, coconut, pepper, etc. In Vallisneria, the case can be studied in the next slide. In the case of coconut and pepper, water is required for the transfer of pollen grains from one flower to another, and also some small insect help for the transfer. In Vallisneria, the male flower reaches above the surface of water and shed the pollen towards water, and female flower is on water surface by water current pollen reaches to stigma. That means male flower expose their pollen grains towards water or shed their pollen grains towards water, and due to water current they reaches to the exposed female flower present on the surface of water. Vallisneria is a aquatic angiosperm. Then wind and water pollinating flower doesn't produce nectar. Normally it doesn't produce nectar, but in some cases they also produce nectars, because nectar is a reward for the pollinating agents like bee, beetle, etc. If the plant have capable of wind and water pollination, so such a reward production is not necessary. Insect pollinating flowers are large, colorful and fragrant, rich in nectar, because they are the rewards, and for attraction they are large and colorful. If the flowers are small, they cluster to form inflorescence for the attraction of insects. For example, Ixora, Techi. Techi means Ixora is a small flower. For the attraction of insects, they join together to form a polycacial cyme inflorescence. Some flowers secrete foul smell to attract beetles, snails, etc. For example, Colocasia, Amorphophallus, etc. The beetles, snails, etc. are the pollinating agents. So they produce a foul smell while when they reaches the reproduction stage. Nectar, pollen grain, and safe place for lay egg, etc. are the rewards for such pollinating agents. So it is a mutual understanding between these flower and pollinating agents. One gain benefit for transfer of pollen, that means flower, and the other get nectar, pollen grain, safer place for egg, etc. Here is a picture. In this picture, a honeybee collect nectar and pollen grain from a sunflower, and also this honeybee is act as a carrier of pollen grain. That means this honeybee is a pollinating agent here. Then economic importance of pollen grains. They are allergen to some peoples. Most of the pollen grains are allergen to some people, not all, but some peoples are allergen. Bee pollen is good immune booster and good medicinal value. It has good medicinal value. Bee pollen can be available as sachet or in bottles. Pollen grain has forensic value for identifying criminals because we studied earlier that pollen grain has a covering called sporopollenin. This sporopollenin can't destroy in any circumstances. So in such a scenario, this pollen grain act as an evidence where that criminal comes or where that criminal goes, or identifying the place from that criminal is come. So it is act as an forensic tool. Then pollen grain provide archaeological evidence due to its resistance to adverse conditions. During excavating archaeological substances, we get several evidences. So pollen grain is also act as an evidence because several thousands of years this sporopollenin can withstand. So we can easily identify uh, that iras flower or plant by identifying this pollen grain. Here there is a bottle called Pumbodi, bee pollen granules. This is an example for pollen grains economic important. Here there are different texture of pollen grains can be seen. Here the ornamentation and different shapes of pollen grains can be identified. And due to this different structure, the pollen taxonomist can easily identify which family belongs to each pollen, so that it gives archaeological as well as forensic evidences. We move to another topic, outbreeding devices. That means continuous self-pollination leads to the formation of inferior quality products. That means plant also prefer cross-pollination. If continuous inbreeding between same plant, that is autogamy or geitonogamy taken place, there is a chance of production of inferior quality product known as inbreeding depression. Note the word again, inbreeding depression. In order to overcome this event, plants prefer cross-pollination except cleistogamous flower, because in the case of cleistogamous flower, the floral parts are not exposed. So there is no scope of cross pollination. So in the case of cleistogamous flower, only the way is self pollination. The various methods adopted by plants to overcome self pollinations are mentioned below. Early development of anther, that means anther developed earlier than pistil or carpel, so that pollen grains shed off. After the shed off, the anther development, sorry, pistil development taken place. So self pollination can be limited up to a limit. Then anther and stigma are at a different position. It is also held for some extent because there is a chance of self pollination here too. And the major outbreeding device is self incompatibility of pollen grain. So note the point, self incompatibility of pollen grain. That means same pollen if reaches to the stigma. That means a flower's same pollen from anther is if reaches to the stigma of that plant, it does not germinate, and that condition is known as self incompatibility. Self incompatibility is the major outbreeding devices that plant shows. Pollen pistil interaction, that means when a pollen reaches to pistil, what changes does it happen? Only the correct pollen grain can be recognized by the stigma. It is genetically mediated. If same pollen reaches to stigma, it can't germinate, called self incompatibility. And another species pollen grain if reaches to stigma, that also can't germinate. So only the correct pollen can germinate. Pollen tube growth taken place towards the ovule, chemotropic. That means we earlier studied that a pollen grain contains two nucleus. One is tube or vegetative nucleus and another is generative nucleus. The function of vegetative nucleus is to produce pollen tube towards ovule, and the ovule produce some chemical signals so that the pollen tube can germinate and move to this ovule, and such a movement is known as chemotropic movement. Through the pollen tube generative nucleus moves. As moving through the tube, it splits into two by mitosis. Then they are called as male gametes, that can discussed in later slides. Then two male gamete reaches the ovule. The way is shown by filiform apparatus or appendages. Filiform appendages is present at the micropylar end. It is connected with synergid. It is a thread-like structure, helps for the way showing structure. That means this filiform apparatus show way towards the ovule opening, that is micropyle. By bursting one of the synergid, two male gametes enter to the embryo sac. That means up to this synergid, the tube nucleus grows. When it reaches to the synergid, one of the synergid, the filiform apparatus show the correct way towards the embryo sac, and by bursting one of the synergy, the pollen tube enters to the embryo sac and releases the two male gametes. Here you can see in the stigma, we earlier discussed about the pistil structure. Here the stigma, the long slender style and ovary can be seen. In this stigma, various pollen grains are reaches. Among them, a few are germinates, and one of them get produced pollen tube towards the ovule, micropylar through micropylar end. That is, this pollen tube moves through the style and reaches to the micropylar end and bursting one of the synergid and releases the two male gamete towards the embryo sac. Here in this picture we can clearly understood the idea. That means a pollen grain, normally a typical pollen grain contain two nucleus. One is vegetative nucleus or tube nucleus and another is generative nucleus. When this pollen tube produces, this tube nucleus guides growth pollen tube towards ovary. That growth is controlled by chemotropism. That chemical is produced by ovary, and through the passage, that means through the tube, the generative nucleus moves. As it moves through the tube, it splits into two and produces two male gametes. Now they are known as two male gametes, and ultimately these two male gametes entered into the embryo sac. These two male gametes entered into the embryo sac. Okay. Artificial hybridization is a technique to breed plants artificially. Here, the first technique is the selection. In selection, superior male and female are selected. We know about Gregor Johann Mendel. He done this technique, that is artificial hybridization, for his study. Then he studied about monohybrid cross, dihybrid cross, etc. Then the second one is emasculation. Emasculation is the major technique in artificial hybridization. Here, if we select two plants and both are bisexual, and so one flower should be treated as male and another should be treated as female. So we must remove the anther from the female plant, because if anther is present in such flower, there is a chance of self pollination. So in order to avoid such a chance, we must remove the anther from the female plant, and that process is known as emasculation. Then artificial pollination or pollen dusting using a brush. From the male plant or male flower, collect the pollen using a brush. Then transfer these to the emasculated flower. That means female flower's stigma. Then we must cover this female flower with a bag, and that process is known as bagging, because there is a chance of pollination from other sources also. That is why we cover this plant, this female plant. That process is called bagging. Then the next stage is tagging. A card is tagged in the female flower in which name of male and female plant, name of person do it, date of doing, so on, etc. are written in that card. It is for future reference. This is a simple sample image of cutting the anther from a female plant. That is, this flower is a bisexual flower, so we must remove the anther from this plant to treat it as a female only plant, and this process is known as emasculation. After emasculation and dusting the pollen grains in the stigma of female plant, we must cover this female plant with a bag, and that polythene bag. That process is known as bagging and then tagging, etc. And after a few days, the fertilization taken place, and fruit, seed, etc. are produced of desirable quality. Up to our desirable quality obtaining, we do the same procedure repeatedly. Then the next stage is fertilization. Fertilization is the second event in the reproduction of flowering plants. Here there are two topics mainly: double fertilization and triple fusion. One of the male gamete fuses with egg, that is first syngamy or fertilization, and form zygote. Another male gamete fuses with the polar nuclei or central cells, that is second fertilization, not syngamy, second fertilization, because syngamy means fusion of male and female gametes. Here one is male gamete and another is polar nuclei, so it is second fertilization. This process is called double fertilization. That means here there is two fertilization taking place. First among one male gamete and an egg, and second among second male gamete and a polar nucleus. So this process is unique to angiosperm only, and this is known as double fertilization. As the second male gamete fuses with two n-conditioned polar nuclei that we discussed earlier, it means a typical embryo sac is 7-celled and 8-nucleated. That means the central polar nuclei is in 2n condition. Two nucleus join together and act as a polar nucleus. The male gamete fuses with this 2n condition. So male gamete n condition and a polar nuclei 2n condition. So a 3n condition is produced. That process is called triple fusion. That means double fertilization and triple fusion are unique phenomenon only present in angiosperm. That means flowering plants. The triploid condition in the center is act as the PEN, that is primary endosperm nucleus, and later it convert as endosperm. So in angiosperm, the PEN or endosperm is triploid in nature, while in the case of gymnosperm The endosperm is haploid in nature. Here we can see three antipodal cells. In the first figure, the two tube nucleus and the generative nucleus are coming contact with the micropolar end, and in the second figure this generative nucleus split as sperm nuclei or male gametes, and tube nucleus burst the one of the synergid and give way for the entering of these two male gametes to the embryo sac, so that these two male gamete now reaches the embryo sac. Then in the figure A, the two male gamete are now reaching to the embryo sac. Here you can see antipodal cells, three numbers, and polar nuclei, 2n condition, not n condition, 2n condition, then two synergids and female gamete, that is egg. In picture B, the two male gametes now reaches to the embryo sac, and in the picture C, first male gamete fuses with female gamete, that is egg, and it is called syngamy, and the second male gamete fuses with secondary nucleus or polar nucleus and produce 3n condition, PEN, that is primary endosperm nucleus. Now in the picture D, you can see the antipodal cells are degenerating. Primary endosperm nucleus produced and oospore, that is zygote, is produced. Now the embryo sac has a zygote and PEN, primary endosperm nucleus. The all other cells degenerates its own. Then we move to the next event, that is post-fertilizational changes. That means in this field we studied about seed, fruit, embryo, endosperm, etc. Endosperm, primary endosperm nucleus, that means 3n condition, later divides and forms endosperm and provide nutrition for the developing embryo. That means the function of primary endosperm nucleus is providing nutrition for developing embryo. The first formed endosperm is nuclear, and later it forms cell wall around the cells and produce cellular endosperm. That means in first condition they are in free state called nuclear form. Then later cell walls are produced around it and form nuclear. It can be seen in this picture, that is first figure A. Free nuclear form can be observed. In the figure B, cell wall is produced around each cells and converted as cellular. Then there are another kind of endosperm, that is helobial. Here both free cellular and cellular structures are combinedly seen. In tender coconut, the endosperm is nuclear. That means the water, tender coconut, that means in elani, etc., the water inside the tender coconut is known as free nuclear endosperm. Later on they get wall formation and aggregated on the shell's corners called coconut kernel. Now that endosperm act as cellular by forming cell walls around the free nucleus. So in tender coconut, the endosperm is cellular, and in the mature coconut, the endosperm is cellular. In some plants, endosperm is completely consumed during embryo development. We know that endosperm provide nutrition for developing tissue, and in some plants the plant completely consume this endosperm. Example, pea, groundnut, etc. And majority of dicot show this pattern. But others, in some others like monocots, they don't consume their endosperm completely during embryo development. That means a few are conserved for future. Example, rice, wheat, barley, etc. and majority of monocot. But some exceptions are there. For example, resinous is a dicot plant, but it show endosperm does not completely consumed. While in the case of orchid is a monocot plant, while it does not have remaining portions of endosperm because the endosperm is completely consumed during embryo development. So we can convert the endosperm as endospermic and non-endospermic seeds. Seed can be converted as endospermic and non-endospermic seeds. In endospermic seeds, most of the monocot, there is a remaining portion of endosperm after embryo development, while in non-endospermic seeds there is no remaining form of endosperm as in the form of starch. Here in the first figure you can see there is a aileron layer. Aileron layer means it is a proteinaceous layer. It is found surrounding the rice, etc. And in the case of dicot seed, the reserve food material, that means proteins, get deposited and stored on the cotyledons. Here you can observe two types of embryos. That the first figure represent the dicot embryo and the second figure represent the monocot embryo. In the first figure you can observe two cotyledons and a radicle and a plumule. Radicle germinates as root and plumule germinates as stem, and two cotyledons present. There is a root cap. While in the case of monocot embryo, it is complex than dicot. It has a sheath surrounding shoot apex and radicle. That is, coleoptile is a sheath that covers the shoot apex, and coleorhiza is a sheath that covers the radicle. And scutellum is the only one cotyledon present in monocot embryo, scutellum. Then the zygote develops in various stages, and some of the stages are mentioned below. Zygote first converted as proembryo, then globular stage, then triangular stage, then heart-shaped stage, then torpedo stage, and finally mature stage. Here the egg, then two cell, then octral, then globular, triangular, heart, torpedo, mature embryo. That means from the zygote, repeated division formation taken place, and finally the mature stage has attained. From this zygote to mature embryo, there are different kinds of stages present. and they are proembryo, globular embryo, triangular shape, heart shape, torpedo stage, etc. In monocot, scutellum is the one cotyledon, and in dicot, the two cotyledon store reserve food in the form of protein. If unfertilized egg develops into an embryo, then it is called parthenogenesis. That means if unfertilized egg develops into an embryo, then it is called parthenogenesis. Example ആണ് honeybee and turkey, etc. In the case of honeybee, the queen is diploid and a drone is haploid. And honeybee queen lay many eggs, and a queen have 32 chromosomes and a drone has 16 chromosomes only. And the laid egg, if drone fertilized, it converted as workers. That means diploid. And the laid egg by the queen laid egg, if not fertilizes, it again converted and develops as embryo, and drone is produced. So in honeybee, both fertilized egg and unfertilized egg taken place. Fertilized egg converted as workers and unfertilized egg converted as drones. So unfertilized egg, it develops into an embryo, it have only haploid chromosomes present. Then we move to seed. A fertilized ovule is known as seed. That means only after fertilization a seed is produced. There are two types, non-albuminous and albuminous. Non-albuminous is otherwise known as non-endospermous, and albuminous is otherwise known as endospermous. In non-albuminous seeds, the endosperm is completely consumed during embryo development that we discussed earlier. And in endospermous seeds, a portion of endosperm is conserved for future use. Example ആണ് rice, wheat. Albuminous seeds example is rice and wheat, and non-albuminous seeds example pea, groundnut, etc. Endospermic versus non-endospermic seeds that we discussed earlier. We studied earlier about nucellus. In black pepper, the ruminate nucellus are also present, called perisperm. In this picture, you can see the perisperm here. That means these perisperms are degraded in most of the fruit, but in the case of some fruits, these perisperms are remain present. That is the remaining portions of nucellus. In seed, the embryo become dormant. When favorable condition occurred, they germinate by imbibition. Imbibition means the absorption of water from the surroundings or from the soil and swell up. It is the property of solid material to absorb water and swell up. Then Lupinus articus. Lupinus articus is a plant seed. It is excavated 10,000 years of dormancy. And Phoenix dactylifera is a seed excavated 2000 years, and both are viable for germination. That means seed dormancy is a period in which that seed is not active but not permanently uh, deactive, but when favorable condition occurs that seed become active. In such a condition, these lupinous articus excavated from Arctic region and they have 10,000 years of dormancy and have still viable effect. And Phoenix dactylifera has 2000 years of viability. Then we move to fruit. A ripened ovary develops into a fruit. In some flowers, the floral parts, thalamus, develop into fruit, and in such cases, they are called false fruit. That means for seed, fertilization is necessary, but in the case of fruit, there is no need of fertilization. That means a mature ovary develops as fruit, while in some cases the thalamus is converted as fleshy edible portion. Such fruits are known as false fruit. Example cashew, apple, etc. In cashew and apple, the thalamus is act as the fleshy edible portion. If fruit is formed without seed, then it is called parthenocarpic fruit. Example banana. In such cases, seed does not produce while fruit formation taken place. That means fruit formed but seed does not produced. Two types of fruits. Here, first one is true fruit and second one is false fruit. In the first case, we can see the pericarp here. That means fruit swollen ovary is converted as the fruit and inside the fruit there is seeds. While in the second case, apple, false fruit, the edible portion is false. That means it is the thalamus portion and the central portion is the pericarp. And pericarp actual fruit is present inside while the edible portion is present outside. Then inside the pericarp there are seeds present. That is why they are known as false fruit. This is an example for parthenocarpic watermelon. Here there is seed-like remnants present, but they are not viable. They are aborted seed. This can be produced by inducing with the applications of hormones. Then we move to the last topics. They are apomixis and polyembryony. Production of seed without fertilization, that is they mimics sexual reproduction. That means we tell about seed formation, that means fertilization is required for seed formation, but there are some abnormal conditions called apomixis. That means production of seed present while fertilization does not take place. That means it mimics sexual reproduction. Mimics means which mistakenly believes that sexual reproduction. The chances are diploid egg produced without reduction division, or one of the cell in embryo sac joined with the male gamete, etc. Actually, we know that one male gamete fuses with the egg and produce zygote, and zygote converted as embryo, etc. While in some plants, the diploid egg is present in some plants. It is an abnormal condition, and that diploid egg directly develops as an embryo. So seed formation taken place, and another case is one of the male gamete fuses with any of the cell in embryo sac. It may be synergid, it may be antipodals, or it may be other cells in the embryo sac and produce egg-like structure and produce embryo. But it is not actual fertilization. It mimics sexual reproduction. Then next one is polyembryony. Polyembryony is the phenomenon in which more than one embryo present in an embryo sac. Here we know that in normally there is one embryo present in an embryo sac. While if we consider in the case of human, there are twins, maybe fraternal or identical, like the same sometime present in plants. That condition is called polyembryony. That means more than one embryo present in plants. While during germination, either one of these embryo get dominance and produce as new plant. Other will be diminished or eliminated. In most of the cases, both are eliminated. In a few cases, one get dominance and other get eliminated. Example is mango and citrus. Mango seeds and citrus seeds generally shows polyembryony. Here is an image. In this image, you can see from a seed itself, from a single seed itself, in the first figure, two embryos. That means two uh, two shoots, means two seedlings in first seed, and three seedlings from a single seed can be produced. That means in the first seed it contain two embryo, and in the second seed it contain three embryos. That conditions is known as polyembryony. Thank you for watching this video. I hope all of you understood the topics that we discussed. We can see in the next class with a new chapter.